Hello and welcome to everyone joining us today. My name's Charlie Carter and I'm delighted to introduce today's session titled Real World Approaches to Sample QC in Single Cell and Nucleus Prep. I'm joined by our speakers, Dr. Christian Urban, the Spatial and Single Cell Sequencing Coordinator at the Functional Genomics Center in Zurich, Andreas Storz, scientist in the Service Lab team at Singleron Biotechnologies, and Songwan Zhou, team leader of the bio team at Logos Biosystems, part of Align Genetics. Please feel free to submit any questions you have during the webinar today. To do this, simply head to the engagement window to the right of the auditorium and click to open the Q&A window. So without further delay, I'm delighted to hand over to our speakers and I'd like to thank them again for presenting to us.
Good morning, good afternoon, and good evening, everyone. Welcome to today's webinar, Real-World Approaches to Sample QC in Single Cell and Nucleus Prep using the LUNA-FX7 Automated Cell Counter. I'm Songwan Zhou from Logos Biosystems, part of Align Genetics, and I'm excited to share practical strategies for Sample QC today. We'll start by briefly introducing myself and today's speakers. I'm the team leader of the bio team at Logos Biosystems, where I focus on developing and optimizing imaging-based solutions for cell analysis. And I'd like to thank our two guest speakers for joining us, Christian Urban from the Functional Genomics Center Zurich, FGCZ, who brings academic expertise, and Andreas Storz from Singleron Biotechnologies, who will share valuable insights from an industry standpoint. Okay, first, let me introduce our company, Align Genetics. It was founded in 2008, 17 years ago. With a team of 75 dedicated employees, we operate in over 115 countries and collaborate with 60 distributors worldwide. We own two brands, Logos Biosystems and GeneScale, and hold 17 patents. And additionally, we have subsidiaries in the USA and France, with our headquarters based in South Korea. Okay, here's today's main focus, the LUNA-FX7 Automated Cell Counter. It offers precise sample counting with reliable size measurements and high-throughput imaging using a variety of cell counting slide options. With both bright-field and fluorescence imaging modes and exceptional autofocusing, the LUNA-FX7 ensures clear and sharp images for accurate QC. The intuitive software also provides flexibility to adapt to diverse sample types. The LUNA-FX7 supports a variety of slide options for different needs. You can select from 1, 2, 3, or 8 channel slides depending on your desired throughput. You can load up to 8 samples, which can be analyzed simultaneously with the 8-channel format. This is a fantastic option for high-throughput analysis using multi-channel pipettes. The 1-channel slide offers the largest sample loading volume of 50 microliters and 47 fields of view, which is great for low concentration samples. The 2-channel slide is the most commonly used option for routine work, while the reusable slide provides a cost-effective and eco-friendly alternative. But most importantly, we now offer the newly developed Spectra Slide AP1, a slide specifically designed for fluorescence-based viability assays. It comes with pre-coded AOPI dyes and includes a built-in sampling button that enables quick pipette-free loading. This slide streamlines the workflow and minimizes variability while delivering convenience and consistency in sample analysis. Okay, then after choosing the appropriate cell counting slide based on what I've described, you can easily load your sample onto your cell counting slide and insert the slide into the LUNA-FX7 and proceed to count and analyze. Then you'll get your counting results. The overall cell counting procedure is quite simple. However, if you're using the Spectra Slide AP1, the workflow is a little different with the other cell counting slides, which requires pipettes. If you're using Spectra Slide AP1, The first thing you need to do is simply pressing the sampling button to activate the internal mechanism. And then dip the tip into your prepared cell suspension. When you release the button, the sample should be drawn into the chamber. And after loading, you just need to insert the slide into the Luna FX7 for analysis, and then you'll get your cell counting results. Okay, so with that, let's welcome our first guest speaker, Christian from FGCZ. He will walk us through his academic perspective on sample QC. And after that, Andreas from Singleron Biotechnologies will offer valuable insights from an industry perspective.
Thank you very much for this nice introduction, Joe. I'm Christian Urban from the Functional Genomics Center Zurich, and I would like to use this opportunity to also warmly welcome everybody to today's webinar. Today I would like to talk to you about sample quality control for single-cell sequencing at the Functional Genomics Center Zurich using the Luna FX7. So first of all, let's get started by briefly talking about the Functional Genomics Center so you can better contextualize the data I'm going to show to you later. We are a multiomics core facility based in Zurich, Switzerland, and we combine genomics, proteomics, metabolomics, biophysics, and bioinformatics in one house, which is great because we don't only generate our own single-cell sequencing data, but we can also analyze it. So thereby we can link together sample input quality and data quality in the end. Functional Genomics Center Zurich is a core facility of ETH Zurich, University of Zurich, and University Hospital of Zurich, but we also have a couple of external customers. So this means we see a vast variety of different species for single-cell experiment, and we also deal with various types of different tissue. We offer a couple of different technologies for single-cell experiments, mainly 10x genomics-based, but we also support BD Rhapsody, Parse Bioscience, Illumina PipSeq, and we always evaluate and try out further technologies. But independent of the technology you are interested in, it is important that you provide a high-quality single-cell suspension once you want to start a single-cell experiment. This means no cell clumps, it should be debris-free, and the cells should have high viability, and obviously you should also have a sufficient number of cells, so you can recover the intended amount of cells. On the right side, you see two images. The left image shows a high-quality single-cell suspension, which is good, and the right one shows a low-quality single-cell suspension, which should be optimized if possible. So there are clumps, debris, low cell viability. All these things create issues later on in the processing workflow and in the data quality. Let's talk about it later on step by step. So sample quality is a very important point of the workflow. We work very closely together with our customers to plan the experiments, and then they generate single-cell suspension and come to us with either fresh or fixed single-cell suspensions. And the first thing we do together is then run a sample quality control, which is a very important point, because if the samples are not meeting the quality recommendations, it might be the best option to stop the experiment at this point and to spend some more time in optimizing single-cell suspension generation. So direct QC before starting and committing thousands of francs or dollars or euros of single-cell reagents is a very important point. For us, QC must fulfill a couple of requirements. So it must work with all the different sample types we are supporting. It must be very fast because we are usually getting samples in big batches, and we need to do QC in batches as well. Obviously, it must be accurate and reproducible, and it should not only be able to count cell numbers, but also to assess viability correctly. We usually deal with very limited single-cell suspensions, so it is important that we are not wasting a large volume on QC. Automated reports make things easy for us to report, but we should also be able to have a look by hand on the samples to assess whether or not we are confident in the results. All these points are actually fulfilled by the Luna FX7, which replaced all our previous cell counters. One of the most important sample quality control parts using a cell counter is obviously the cell count. So we really need accurate and precise and reproducible cell counts when running single cell experiments. This is because you want to neither overload nor underload your single cell experiments. If you overload, you create an increased number of multiplets in your single cell experiment, which can impact your data quality later on. If you underload, you are getting fewer data points for the same experimental price. But also very important, accurate cell counts are already important when combining your single cell suspensions if you would like to use cell multiplexing. For example, at the bottom left, you can see that 3 samples are being multiplexed by, for example, cell hashing with differently tagged antibodies. And then all 3 samples are combined in one pool, which is then loaded in one single cell experiment. So if you're off with the cell counts between these samples, you're creating an imbalance of your final pool, thereby over-representing or under-representing certain samples, which is later on not good for your bioinformatics analysis. Precise cell numbers are important, as mentioned. And it is very important to know that most of our customers come to us after actually having cell numbers counted by a fax instrument. What we see here is usually a big discrepancy of about 50% to the cell numbers we estimate with the Luna FX7. Fax facilities usually give up to 50% more higher cell numbers than we later on count at our facility. So obviously you now are wondering which number is the correct number. So I went back to reassess all the experiments we did over the past few months and compare actually recovered cells after the experiment and the analysis in comparison to the number of cells we intended to recover. So this is shown in the plot you see at the bottom right area for both fresh and PFA fixed samples. So as you can see, we loaded these experiments using the data we get from the Luna FX7. We actually recovered consistently the intended cell number for both fresh samples and PFA fixed samples. Just please be aware that if you're working with PFA fixed samples, the fixation causes your cells to shrink a little bit, and therefore you might need to actually lower the cutoff for cell size when you are doing cell counts for PFA fixed samples. The second quality controller aspect I would like to touch on is cell viability. This is very important to generate later on good quality data for analysis. Because if you have a low viability, first of all, you have a lot of dead cells included in your experiment, which need to be removed later on, and are actually lowering the number of cells you have included in your analysis. But also dying cells are releasing RNA to the buffer they are in, which means you get a higher percentage of ambient RNA in your buffer. Especially if you're working with droplet-based single-cell methods, these can later on result in a high background of ambient RNA in your whole data set, thereby compromising your final data. In addition, you need to consider debris linked or RNA which is stuck to debris pieces. So if you have high amount of debris present in your sample, these could end up in those droplets and carry a bit of RNA in there and then misinterpreted as actual cells in the final data analysis. All these data can then be reanalyzed using bioinformatic methods, and thereby you can try to remove the background, but it is best to start with a high quality single cell suspension to make sure this is not even necessary. Single cell workflow optimization is a crucial part of most of the single cell experiments being conducted. On the right side, you see an exemplary workflow of a complex single-cell experiment, starting with tissue dissociation, going down to the actual single-cell experiment. So it is always good to have a couple of QC points, especially before committing pricey reagents, to make sure that you are still having a high viability single-cell suspension, and you still have sufficient numbers of cells present. All these different steps in this workflow can put stress on your cells in the suspension, and can actually, by washing and filtration steps, decrease the cell number you have in your suspension. So if you don't want to be surprised by the final QC, it's good to have a couple of QC points included in your workflow. This is also especially important right after tissue dissociation, because here you really want to see how your cell viability looks like, especially if you are removing dead cells later on via FACS sorting. Why is that? If you want to get a broad overview about cells included in your single-cell suspension, you might have some which are more fragile and some which are more robust. So right after you do the tissue dissociation, it's good to check your single-cell suspension before enriching for viable cells, because you might actually decrease incorporate a bias by doing that. All of your fragile cells might have already been dead at this point, and it's important to check whether or not the viability looks good after dissociation. Then, if you have any pricey steps, like for example cell surface protein staining with an antibody cocktail, it's also good to check suspension quality right before doing such pricey steps. And obviously, the last sample QC after you stressed your cells with all these steps in the workflow right before committing single-cell reagents to your experiment is very important to decide whether or not to move forward or to actually re-optimize the workflow. With this, I would like to summarize that reliable sample QC is important for every single-cell experiment. Especially the last QC point before committing expensive single-cell reagents is important. But it is also important during all the optimization steps, which are usually included in setting up each single-cell experiment. With this, I would like to thank you all very much for your attention, and I'm going to hand over to Andreas now, who's going to talk about single-cell sequencing and quality control from a company's perspective.
Hello everyone, also from my side. My name is Andreas Storz and I'm a scientist in the service lab team at Singleron Biotechnologies, where I'm based in Cologne in Germany. There, as a team leader, I ensure our day-to-day handling of the customer clinical and research samples in our labs. This includes, for example, tissue dissociation, cell counting, and the preparation from single-cell NGS libraries, which are sent out for sequencing. Before I start with an insight on how LunaFX7 plays a crucial part in our workflow, I would like to shortly introduce our company. So, Singleron Biotechnology is a molecular diagnostic company with a focus on single-cell sequencing techniques. The company was founded in China in 2018, and from there we rapidly expanded also to the other continents, to Europe, Singapore, and in the US. And for example, in Europe, where I'm working, in Cologne, we also have established offices, labs, and manufacturing facilities. So what is our mission at Singleron? So the mission is to find out what is the role of certain cell types in different contexts or on different layers. So our task is to apply single-cell analysis techniques in clinical diagnosis, drug development, and health management. Our organs, or tissues of any species, have a high diversity of cell types, and we are able to investigate those on multiple layers, like the different cell types, what is their role in specific disease models, or also how is the gene expression level in certain cell types affected or is changed, or we can also investigate cell-to-cell interactions. We do this like on the basis of a unique microfluidic chip, and on the basis of this microfluidic chip, we offer complete solution for single-cell sequencing technologies to clinicians and researchers, both as a kit or like a service solution. Our company's knowledge also includes like bioinformatics, instrument design, and clinical product development. Our dissociation and processing with a microfluidic chip can also be performed both manual or automated with devices like the Python for tissue dissociation or Matrix Neo for chip processing. A picture of these devices you can see in the upper right corner of the slide. On this slide, I want to give you like an overview more what is like happening in our service lab and how is our workflow look like. And also I want to show where Luna is a part of. As you can see, we have like several QC steps in our workflow until we release a bioinformatics report to our customer. Our wet lab processing part starts with the tissue dissociation from samples that were sent by customers. And after we finished a cell suspension QC, the cells are loaded on the microfluidic chip for partitioning. For this, I want to show you that figure to shortly explain how like the microfluidic chip is working. So in this picture you can see what is happening. So the standard version of our chip has up to 150,000 microvels, and each microvel can capture one single cell. And to capture like the mRNA, we add specified barcoding beads, which are designed in a way to bind the mRNA over a poly-T ending attached to a barcode sequence. And after adding a lysis buffer with the beads and the cells already loaded, the cells are lysed, the cellular mRNA is released and is also captured by barcode beads. And in this way, every single cell has like its own specific barcode bead. And after a bead retrieval, we perform a reverse transcription and amplify this to a cDNA from which we can construct NGS libraries, which are then sent out for sequencing. This is an overview of the complete workflow, and as you might guess, is that Luna is of course like in our in the process of the tissue dissociation and cell suspension QC a large game changer for us. We at Singleron also have like a wide experience of tissue and sample types, which we process like fresh for a single cell suspension, or when also it's suited for the project, or some like tissue types like muscle, for example, which are suited better to be dissociated in a frozen state for single nuclei suspension. We also had like tissues already from monkey, fish, fruit fly, or plants. This is about our mission. On the next slide, I want to point out what challenges you can encounter during tissue dissociation or when you want to count a cell suspension. The expectation is a cell suspension that is shown like in the pictures above. What we can observe here is clearly separated cells, 100% viability, and no background at all. This sounds like a walk in the park for researchers, but the reality is shown in the pictures below. Usually cell suspensions from tissue have a lot of background. They have dead cells, debris, or red blood cells that needs to be removed at certain cleaning steps or washing steps. Or also it can also happen that you have no clearly visible nuclei when you process frozen tissue. In addition to that, it is also mostly another challenge is that the starting material that is provided by the customer is mostly limited, especially when it's, for example, a patient sample. On top of that, the cells also needs to be viable until the end of the workflow. That means after washing steps or removal step, the cell suspension needs to be counted many times, which can also be indeed very time consuming. And on top of that, an accurate cell suspension QC and count is crucial for every success of the project. And at this point, Luna FS7 with its automated counting is a big game changer for everyone who works with cells. On the next slides, I want to show you an example for both a single cell and a single nuclei project. This slide is about a clinical trial project we had in our company from samples that were from human intestine. The sample nature was like that the cell suspension was mostly very clumpy, and also we saw some aggregates, which made it very difficult also to have an accurate count. The aim of the project was like to capture every time 6,000 to 7,000 cells. And especially here in this project, it shows that like consistency is very important for clinical trials, because in the lab there can be a change in the operator, and also the patient sample is different every time. Despite optimizing the tissue dissociation, we also established with LUNA-FX7 and its several parameter function and dilution calculator a counting protocol that was delivering consistent accurate cell counts which were independent from operator and sample. And this you can see very nicely in the chart, where we can see the number of the cells that have been captured for 27 samples in total, on different time points and also different patient samples. And on the right by the chart, you can see a result of one of our sequencing results with the estimated number of cells that have been captured on top. And also a deciding factor here is the mean reads per cell. The thing is that our customers are paying by reads per cell, and if there would be every time too much difference in the cell counts or the cell capture rate, the reads per cell would drop, and this would also give us, like in the downstream sequencing data, a poor outcome. My second example shown on this slide is about single nuclei. In this slide we see nuclei suspension pictures that were like made from when we dissociated frozen mouse brain. With the AOPI staining, we can easily distinguish nuclei from living cells due to the red fluorescence from the PI count. But here, in this slide, I want to point out how good Luna is performing for nuclei suspension, which also have high debris content. The upper pictures are with and without debris removal, and the lower with debris removal. And a comparison was also processed on our microfluidic chips, where you can see the sequencing data for both on the right side. And as we can see, for both chips, like a similar number of nuclei were captured. It was like around 6,500, and the target was 6,000 to 7,000. Of course, it is not advisable to proceed with debris-rich samples, but sometimes the number of the extracted nuclei from the limited starting material is so low that a removal step is not always feasible. And the main difference we can see at the sequence, because the fraction read in cells is of course much lower in the debris-rich sample due to the ambient RNA of the debris in the background. This was my examples for single cell and single nuclei. Slide, I want to summarize like this part of the webinar. So some key challenges we also have with customers who buy our kits is, for example, that sometimes the transition for counting like from manual to automated system can be met with resistance, because there is like sometimes a very strong reliance on fax-based or manual counting methods, and it's also believed to be that cell counting is very time consuming. Manual counting is also very time consuming, and there is like some concerns about the cell viability when you use too much time for cell counting. And that's why our message is that automated QC for us is not optional, it is very essential, because as you could see like in the previous slides, that an inaccurate or inconsistent cell suspension count can jeopardize the entire workflow up to downstream processes like sequencing. And also, especially for clinical trials, a proper QC of the suspension ensures like consistent data quality, reproducibility, and also the success of the project. For example, when we would have like underestimated cell counts, or then it could result in too less cDNA, too less captured or too much captured cells, and this would result in very low reads per cell, culminating in poor sequencing data outcome. That was from my side. On the right corner, you can also see the address of our webpage, where you can find some details about our products and what we offer. And now I want to give back to Joe, who will talk about how Lunar FX7 will work, which is like indeed a very great and reliable product. And thank you.
Thank you, Christian and Andres, for the insightful presentations from both academic and industry perspectives. Now I'll walk you through how the Luna FX7 enables practical sample quality control by showing real-world examples across various sample types. Okay, to start, here's the main focus of this session: how the Luna FX7 enables image-based sample quality control. By using dual staining, we can clearly distinguish between viable cells, non-viable cells, and isolated nuclei based on fluorescence intensity and membrane integrity. This fluorescence-based classification allows the system to accurately identify target objects while effectively filtering out debris and artifacts. We have successfully applied this workflow across a range of real-world samples, such as fresh, frozen, and FFPE tissue samples. And you might be wondering why we strongly recommend fluorescence staining, such as acridine orange and propylium iodide, AOPI staining, over traditional bright field dyes like trypan blue, TB. While the Luna FX7 does support bright field imaging, including TB, there are important limitations to consider. In this slide, we compare nuclei samples stained with AOPI and TB to show the difference in performance, especially in debris-rich samples like isolated nuclei. Trypan blue tends to stain both nuclei and surrounding debris, and this can lead to false positives. It can also fail to stain some nuclei. This results in false negatives. And ultimately, this can compromise both count accuracy and viability assessment. In contrast, AOPI staining offers much higher specificity. AO stains viable whole cells green, and this signals should be absent in isolated nuclei I'm showing here, while PI stains nuclei red. Combined with the Luna FX7's imaging and classification algorithms, this enables accurate identification of target objects and effective exclusion of debris. With that, let's jump right into the fluorescence cell counting using the Luna FX7. So if you select this option on the Luna FX7, you'll see two different modes available. One is cell lines and primary cells on the left, and another one on the right is cell lines and primary cells advanced. For clarity, we refer to these as basing mode and advanced mode. Both modes provide comparable performance in fluorescence detection and cell counting. However, the main difference is declumping performance. Advanced mode offers better declumping performance, which makes it effective for samples with aggregated cells or clustered nuclei. And as you probably know, the sample preparation steps for isolating cells and nuclei from tissues are often harsh on samples. And this can yield high levels of clumped target objects. Therefore, we recommend using advanced mode for more accurate analysis. And if you select advanced mode, you'll have access to protocol tab at the top of the screen. This is where you can fine-tune key imaging parameters to match the specific characteristics of your sample. And what makes the LUNA-FX7 especially powerful is its protocol flexibility. You can easily modify, save, or load settings optimized to different sample types. And as you can see in this screen, there are customizable settings, including GF and RF exposure levels to adjust fluorescence intensity, minimum and maximum search size, which helps define the object size range for detection. And this is especially useful for filtering out debris or any large aggregates. Declumping sensitivity, this is where higher values improve separation of clustered cells or nuclei. Minimum fluorescence intensity is for excluding dim or low signal particles. And minimum roundness, we generally don't recommend adjusting this as it only has limited impact. And you can also enter a dilution factor if needed to ensure accurate cell concentration calculations. So the example shown in this screen uses the default protocol. This is optimized for general fluorescence-based viability assays. And all samples we'll show in this webinar were analyzed using this default protocol. And the viability assays using the advanced mode must be used with fluorescence staining. We recommend using AOPI staining for accurate sample QC in both cells and nuclei. The staining principle behind using AOPI is membrane permeability. AO, or accurate in orange, is a membrane permeable dye that stains the nucleus of viable intact cells, emitting a green fluorescence signal. PI, or propydium iodide, on the other hand, is red membrane impermeable dye. It only enters dead cells or isolated nuclei where the membrane is compromised or absent. An important aspect of this staining method is the fluorescence quenching effect. When both dyes are bound to cell nuclei, the PI signal absorbs the AO signal. This minimizes bleed-through and prevents false double positive results with both red and green signals present. On the right, you can see visual examples. Intact cells appear green, dead cells show red signal due to membrane damage, and isolated nuclei, which does not have membrane, also stain red. This dual staining approach allows for precise classification and supports reliable image-based QC. And using AOPI staining is quite straightforward. You can simply mix your fluorescent dyes with your cell samples in a 1:9 ratio. And you can load your samples onto a cell counting slide and count the sample using the LUNA-FX7 using the default protocol. On the left, you can see a montage of cell samples with approximately 50% viability. Live cells are stained green by accurate in orange, while dead cells appear red due to propydium iodide uptake. As I mentioned earlier, the quenching effect allows the red PI signal to override the green AO signal in non-viable cells. And this makes it easy to visually distinguish between live and dead populations. Okay, and let's say you have stained your cells and you have your analysis results. then there are several important factors to consider when evaluating the quality of your samples under fluorescence-based analysis. In the montage provided on the left, you can see bright field of fluorescent and tagged images captured after analysis. The green circle indicates live intact cells. These cells should be disregarded when assessing isolated nuclei. The red circle highlights dead cells or nuclei, and it's important to observe the overall morphology. So basically, checking if the cytoplasm is completely removed or not from nuclei is very important. Successfully isolated nuclei typically have an elliptical shape, while damaged nuclei may appear irregular. Debris is not a concern because it won't emit a fluorescent signal, meaning any debris won't be tagged as objects by the algorithm. Okay, knowing these criteria and staining principles, cells and nuclei should show distinct characteristics. In the bright field images, it should be easy to distinguish between intact cells and nuclei. And this allows users to ensure there's no remaining cytoplasm or irregularly shaped nuclei. In the fluorescent images, green signals should only be visible from intact cells, while red signals should only originate from isolated nuclei. Moving to tagged images, you will observe a significant change in viability. Okay, with that in mind, let's take a close look at some real-world samples. And here, we are looking at examples of isolated cells, first, from mouse spleen and blood. Specifically, mouse splenocytes on the left and PBMCs on the right. Both samples were stained using AOPI and analyzed on the LUNA FX7. Green signals represent viable intact cells stained with AO, while red signals represent non-viable cells stained with PI. At the bottom of the slide, you can see magnified views of the selected regions. And this close-up shows how clearly the LUNA FX7 can distinguish between live and dead cells, even in samples with mixed populations. However, isolated cells can vary widely in size. While larger cells, typically over 10 micrometers, are generally easy to analyze, many isolated cells are smaller than standard mammalian cell lines, and this can make image-based analysis more challenging. To address any concerns, here's a montage comparing AOPI staining performance between U937 cells, which is around 14 micrometers, and PBMCs, which are generally smaller than 10 micrometers. The samples represent a full range of theoretical viabilities from 0% to 100%. And as you can see, the green to red fluorescence ratio shifts accordingly. And this confirms that AOPI staining provides a consistent and reliable viability assessment, even for small cell types. Now that we have shown isolated cells, let's get into nuclei samples isolated from tissues. These are examples of isolated nuclei from fresh tissue samples, including mouse brain and heart. All samples were analyzed using the AOPI staining paired with the LUNA FX7. Each panel shows a full field of view at the top and a zoomed-in view below, with red circles indicating nuclei tagged by the system. Despite differences in sample quality and debris content, the LUNA FX7 consistently detects intact nuclei thanks to its advanced imaging and algorithms. And next, let's take a look at nuclei from frozen tissue samples. including mouse lung, kidney, liver, and small intestine. Again, all samples were stained with AOPI and analyzed using the LUNA-FX7. And you can see full images on the top and zoomed in views below, with red circles marking nuclei detected by the system. Frozen tissues can be challenging due to variable ice crystal damage or partial degradation. But the LUNA-FX7 can still perform reliably. It could detect intact nuclei across diverse tissue types. Finally, here are nuclei isolated from FFPE tissue samples, mouse kidney and spleen. FFPE samples are more difficult to work with compared to fresh and frozen tissues. Formalin fixation and paraffin embedding may result in protein cross-linking and elevated debris levels. These factors require additional care for more accurate analysis. On the left, you will notice objects tagged with green circles. And these are not intact live cells, as FFPE samples should not contain any viable cells. These false positive signals are likely the result of nonspecific AO binding to debris. And that's why it's important to carefully interpret your results, and in some cases, consider single staining strategies to improve accuracy. I'll walk you through key considerations for handling samples like FFPE, along with common issues for all sample types. Here is a montage shows a comparison between nuclei isolated from fresh kidney and FFPE kidney samples. As many of you know, preparing FFPE samples requires additional steps such as paraffin removal and rehydration using graded alcohols. And these steps can compromise overall sample quality. A common issue with FFPE samples is the nonspecific binding of acridin orange to debris. This debris tends to nonspecifically bind to acridin orange, AO, and this can produce misleading green fluorescence that does not reflect viable or intact cells. Importantly, there should be no live intact cells present in the sample, since these tissues have been fixed. To address this, we recommend either simply ignoring green signals entirely when working with FFPE nuclei, or for better specificity, using single staining with membrane impermeable dyes like propylium iodide, PI, alone. This approach helps reduce background noise and improves accuracy when analyzing FFPE-derived nuclei. And another common challenge with FFPE samples is limited dye penetration into the nuclei, caused by formalin-induced cross-linking. This often results in weaker fluorescence signals, especially when using membrane-impermeable dyes like propylium iodide, PI. To address this, we recommend increasing the RF LED intensity from level 5 to 7. This adjustment enhances signal strength and improves detection accuracy during image analysis. This helps ensure more reliable staining and analysis results. And these were two key issues specific to FFPE-derived nuclei. Now, let's move on to more common challenges that can affect all sample types. One of the most common challenges in sample preparation is debris. The process of isolating cells or nuclei is often harsh and complex, which can lead to high debris levels in the final sample. We generally do not recommend using samples with high debris levels, but the LUNA-FX7 can still handle them. If you look at the image on the left, it shows a sample before filtering during the nuclei isolation process. Even under these challenging conditions, the LUNA-FX7 combined with AOPI staining can still accurately detect the target objects. However, it's still important to interpret your results with caution, as debris can introduce background noise that may affect your analysis. Therefore, I would recommend to remove debris as much as possible. If you're dealing with high debris samples, filtering and washing are critical steps for reducing background noise and improving overall quality. As you can see in this montage, these steps can significantly improve clarity. However, excessive washing steps can compromise sample integrity. That can lead to aggregation and a lower recovery of target objects. And that's why it's important to balance between debris removal and sample preservation to maintain both data quality and sample yield for any types of downstream applications. And here's another common challenge, which is achieving the correct focal plane. While the LUNA-FX7 has a highly reliable autofocusing algorithm, smaller objects such as nuclei or immune cells in the 5 to 8 micrometer range may take longer to fully settle. If imaging begins too early, these objects may appear out of focus, potentially impacting your results. To ensure optimal focus and accurate analysis, we recommend waiting up to 2 minutes after loading the sample to allow it to fully settle in the chamber. This simple step can significantly improve focus accuracy, especially when analyzing small or delicate targets. Please take a moment to check your sample and allow adequate settling time before imaging. It's a quick but very effective way to improve the reliability of your results. And that wraps up today's webinar. In conclusion, both single-cell and nucleus isolation is a time-consuming and delicate processes, and ensuring good quality control is essential for downstream applications. Automated cell counters like the LUNA-FX7 can offer a straightforward solution for sample QC. For more information, please visit our website at logosbio.com, where you'll find detailed product information, application and technical notes, instructional videos, and webinars. And thank you very much for joining today's webinar.
Excellent. Thank you all for your insights today. It's now time for our question and answer session. So just a reminder to everyone listening in, if you would like to ask a question, you just need to simply head to the same engagement window to the right of the auditorium and click to open your Q&A window. So to kick off the final portion of today's event, the first question I have is, how do your QC results on the FX7 correlate with downstream sequencing outcomes? Christian, if you wouldn't mind starting.
Thank you, Charlie. Yes, you briefly saw in the talk from Andreas and me that basically cell counts are very consistent. So cell recovery is very predictive based on the cell number from the LUNA-FX7. What I didn't touch on in the presentation is dead cells, so viability, and the resulting mRNA, the resulting ambient RNA levels. So this also correlates really well. So it depends a bit on the cell type, but basically the number of cells, dead cells removed, afterwards in the analysis correlates really well with the viability assessment of the instrument.
Lovely. Thank you, Christian. Andreas, would you like to add anything?
No, I think that's like everything what we can say to that. I mean, as Christian say, for the downstream sequencing results, it's like very important to have a precise cell count, like because the sequencing is also like a very expensive part of the workflow, and therefore it's like also huge a good advice like to do the cell counting in a good way. This can also save you a lot of money, and especially for the sequencing results, especially for the ambient RNA. It is also good to provide clean cell suspension and precise counted cell suspension. Thank you.
Excellent. Thank you. That was great, Andreas. Our next question is for samples with a high background fluorescence or autofluorescence. How have you optimized the LED intensity or dye selection for a better signal clarity on the FX7? Maybe one for you, Joe.
Yeah, that's a really important question, especially for users working with complex samples. On the Luna FX7, you can fine tune the LED intensity settings for each fluorescence channel, which is critical when dealing with high background or autofluorescent samples. Um, in practice, we recommend starting with default protocol with LED levels of 5 and 5, and gradually increasing it while or decreasing it while monitoring the signal to background ratio. This helps avoid saturating the detector or amplifying background noise.
Lovely. Thank you so much, Joe. Unfortunately, that is all we have time for today. So I'd like to extend a huge thank you to the three of you for your discussion and presentation and for answering the questions that we had time for. And thank you as well to everyone who's joined us online and to those who submitted a question. We hope that you found this a worthwhile session. If we didn't quite get to your question today, we will follow up with answers to these shortly, or if you have any questions post-event, please feel free to email us at editor@selectscience.net. If you're currently attending the live webinar, you'll automatically receive a certificate of attendance, including the learning outcomes summary for your continuing education purposes. But if you're listening to this on demand, you can request the certificate by emailing editor@selectscience.net. If you'd like to listen again to today's webinar or invite a colleague to listen, it will be available on demand in just a few days' time. So thank you all once again for joining.